Okay, let's start this webinar session. So, hi everyone. I'm really glad to be the host of this webinar. I'm Alban. I'm the CEO of Immunosmol, and along with Dominique here, our antibody development director, just here. We have been developing since more than 50 years now antibodies detecting small molecules such as amino acids, their metabolites, toxins. And so, Dominique.
Hello. So, welcome to you all, and I'm really happy today to listen to Dr. Hei Ming LAI. In his latest paper, he and his team wanted to know if the highly multiplexed approach 3D histology could be compatible with neurotransmitter imaging. So they test antibodies against dopamine, serotonin, GABA, glutamate, glycine, and noradrenaline. So they all work fine without any exception. So it's a great pleasure for us to see that the tool we are developed could be useful to great scientists.
Perfect. So we are on the same page here. So now, before jumping to the presentation, just a quick introduction. So Hei Ming LAI was trained as a medical doctor specialized in pathology at the Chinese University of Hong Kong. He is now a clinical assistant professor at the Department of Chemical Pathology at the same university. And over the past decade, he and his team have been developing novel 3D histology method for clinical and research applications. These works have been published in nice and prestigious journals such as Nature Method, Nature Communications, Neuropathology and Applied Neurobiology. And their technology were also recognized by the 3 gold medals in the International Exhibition of Inventions in Geneva. Their latest technology, INSIHGT, for in situ host-guest chemistry for 3-dimensional histopathology, can achieve uniform, deeply penetrating multiplexed immunohistochemistry in centimetre-scale samples. And it allows to do spatial transcriptomics with mRNA detection, but also, as Dominique mentioned, can allow detection of neurotransmitters, so dopamine, serotonin, etc. Can allow also post-translational and epigenetic modification, evaluation, and everything in 3D. So, Dr. LAI, the floor is yours.
Thank you. Thank you, Immunosmol team, for kindly inviting me. And it's also a privilege to be invited. So, yeah, so I'm glad to see you all. So perhaps we can talk a little bit about 3D imaging and just perhaps enjoy the beautiful images we usually see day by day. So I can share my screen. Let's see. It's kind of, yeah. So I presume everyone can see my screen, or if there is any problem or any questions burning, just feel free to ask anytime. So yeah, today here I'm trying to introduce to you our technology, which I believe might benefit your research very easily, and especially with antibodies like those from Immunosmol that you can use to probe any kind of things from histone modification, neurotransmitters, small molecules to proteins. And it is a very easy to use method, and hopefully I will be able to convince you that allows you to get good images. So I will talk a little bit about how we do deep immunohistochemistry and how we analyze the whole tissue. And we'll also go over some application examples. So hopefully that will be of interest to you. So my journey started when I was still an undergraduate as a medical student. So at that time, my kind of undergraduate supervisor, he would do this kind of neuroanatomy, like cutting all the mouse spinal cord very, very tediously onto glass slides. So including even the bits of the sacrospinal cord. But when I joined the lab, there is already quite good technology coming up that you can visualize fluorescently tagged mouse spinal cords. So at that time already, I thought the whole problem is solved. So you can really trace the spinal cord injury, nerve connections very easily. But the problem is, when I try to apply the techniques for my own research, and you can really get a tissue very clear, but you can never try to get the probes deep inside the tissue. So even if it is transparent, you have to stain it with something else to visualize the molecules or the cells that you need to see, even with the most powerful microscope. So I come to realize that the deep immunohistochemistry is really the challenge. So you have to first get the probe not only superficially, but also uniformly, not to get your data biased. Like getting a ground truth data, it's like marinating an egg, but you have to get it fast and also homogeneously. So sometimes, for example, you will see this kind of brain that only the stain will penetrate on the outside, like the pink area here, but not like the ground truth here in the green channel. So over the last couple of years, I think like 3 to 4 years, there have been a lot of different methods coming out. They are all very, very innovative, trying to get antibodies easily into the tissue. So for example, cubic histovision is published by the prestigious Japanese group. They use high salts and also some other chemicals that when you marinate the brain, a very permeabilized brain for like several weeks, then you can get the antibodies deep inside the tissue. Or you can just turn the tissue into an elastic gel. You can stretch it thin so you can push the antibody in mechanically. And there are other groups uses electric field, or if you are lucky enough, you have your tissue comes with blood vessels, you can just stick a pipe in there and perfuse all the stains there. And previously, we tried to use temperature to push thermostabilized antibodies into the tissue. But all these kind of methods have their drawbacks. For example, these are quite tedious or not applicable to all tissues. Cubic histovision is a little bit too long and too slow. And sometimes spears will cause some kind of non-specific staining. So we come to realize that if we want to develop a good method, the researchers always want to use antibodies that they develop in the lab or buy from the vendors. They will want to have their compatibility with any kind of tissues, organoids, embryos, cancer, clinical samples, archive tissues, specialized fixative, something like that. And they will always want more colors. They never get enough of the colors. And they will always want faster methods, but simpler procedures, and also, of course, lower, a cheaper procedure. So I think this kind of drives us to improve our method further. So we wonder whether we can have as good scope as ELAS. For example, ELAS can process like a whole human brain slab. So it's unlimited in size, but optimized for speed, like E-flash and perfusion that is only taking 1 day of staining, and also optimized for simplicity, so that you don't need any specialized equipment. You don't need to buy a machine to make electric fields or something like that. And we finally developed INSIHGT that fulfills all these criteria. You don't need a specialized equipment for that one. You don't need a special fixative for that. It's also the faster techniques you can get. And we tested 323 antibodies and counting, off-the-shelf antibodies. You don't need primary conjugated antibodies. Just primary antibody is okay. So the way it works that we can try to explain a little bit how deep immunostaining is so difficult. So when we apply antibodies to the tissue, if we see on the left side that you will have all these antigens on the surface, it will kind of immediately trap all the antibodies there. So they can't move further. They have to bind to their targets. So what we did with INSIHGT is actually an acronym for In Situ Host-Guest chemistry for 3D histology, is that we have a chemical inhibitor that we mix with the antibody, but it would just only inhibit any protein-protein interaction, but do not denature them. So they can diffuse homogeneously and distribute evenly across the whole tissue. And after that, we will complex away the inhibitor so they can bind to the deeply located antigens as expected. So now you will get a whole uniform immunostaining. And the host-guest chemistry is actually based on this new chemistry we discovered called the boron cluster compound. So here is the cluster compound called B12H12. You call it dodecaborate. It's a host. And when it gets complexed by a cyclodextrin, then its activity of inhibiting protein-protein interaction will be removed. So that this inhibition will cause the antibody to bind to the antigen at any space in time. So it's kind of similar to a previously published method called switch, but switch uses SDS, and it often causes unpredictable non-specific staining. For example, sometimes it just fails to stain at all. So the detergent is obviously denaturing the antibody. Sometimes it will just create these kind of vascular precipitates, or even the wrong staining, like GFAP labeling the neurons. And even if you change the fixative, it won't help in all cases. So there is no one-size-fits-all protocol. But INSIGHT using this kind of system is kind of unconventional. It's not a detergent. Then you can get all the predicted staining in all kinds of tissues for all antigens. And of course, we went ahead and test 300 of them because the technique is very simple. It's like doing an ELISA. So you can just do it on the 96 well plate, and as simple as that one. And also it's the same protocol. So you don't have to optimize different fixation, blocking, permeabilization steps. It's just all the same protocol for all of the samples that we have imaged from day 1 to today for about 1.5 years already. And it's also the fastest. If we count for a whole mouse brain, you have a fixed tissue to image in 6 days. And that's equivalently for a 1 cm cube tissue. You can also do it within 6 days. So for example, here we benchmark iDisco, which is quite a popular whole brain imaging technique. I scan through of the whole mouse brain with INSIGHT. So INSIGHT, actually the procedure is following iDisco. So nothing has changed except when the staining step was have this kind of chemical added. So you can see the effect is quite dramatic. So in the middle slice, you can see that only the for iDisco only the outside is stained, but with shorter staining time, INSIGHT can even achieve a greater penetration, and you can even see the substantial number dopaminergic neurons here. So it's quite a dramatic enhancement in uniformity and also depth of staining. And of course, we do some quantification. We have a device scheme. So for example, we have this hemi mouse brain. So we cut the brain in half, and we first stain it with whatever deep staining method you have in magenta. And then we cut it open at the thickest dimension, and then we stain it. We call it cut staining with just conventional PBS and twin 20. And then we try to image on the cut plane to see whether there is perfect correlation with the bulk staining channel versus the cut staining channel. Ideally, they should be the same. So here is the results. So you can see the INSIGHT almost have all the overlaps in terms of the magenta and also the green one, but others still suffer from a little bit of the inhomogeneity. So if we quantify each of the dots, each of the cells there, then you can get this kind of exponential decay as we get deeper into the tissue in terms of penetration distance. But for INSIGHT, the green line one is almost flat. It's almost ideal. So it is independent of the penetration distance. And that is what we are looking for. And in fact, if we correlate back the reference signal with the INSIGHT signal, you get nearly perfect correlation linearly. So it kind of provides you a semi-quantitative measurements of the relative protein expression in each cell across the whole 3D space. And of course, we want to see whether the delicate structures or the molecules is retained in the tissue. So you can even use antibodies to probe mono, di, or trimethylation. You can probe acetylation. You will see different kind of immunostaining patterns, verifying the staining is correct. You can even distinguish the symmetricity of the arginine methylation, whether it's located on one nitrogen or distributed across 2 nitrogens. So the symmetric case is only located outside of the seminiferous tubules. And of course, for epigenetics is useful for human cancer like glioma. We will measure the trimethylation. So that is clinically useful. And of course, with immunosomes, neurotransmitter antibodies, we also try to do our dreams of mapping the neurotransmitter in the brain. So we tested GABA, L-glutamate, D-glutamate, serotonin, histamine, noradrenaline, and dopamine. So these are the standard neurotransmitter. And we are trying to map the whole brain in terms of these neurotransmitter as a side project. So because it's is fun because it provides information that is unmatched by proteins. And of course, you can scale it up. So you have this human cerebellum. If you want to stain a larger chunk of tissue, all you need to do is to prolong your staining time, and the washing time is just the same. It's still the fastest method, and the details are still preserved at the highest resolution level. You can go even further to centimeter scales in 10 days, and all these kind of vessel staining is still uniform, and like there is no rimming of the signal on the outside of the tissue. So this one is actually so large that we can't put into our light sheet microscope. We have to cut it open at the thickest dimension and then image it with confocal. So that would kind of allow of some unusual approaches to analyze the tissue. For example, here we have a Parkinson's disease brain. So we take out the whole human brain and we cut out a half of the brain stem and we map the phosphorylation on the alpha-synuclein to see the Lewy pathologies across the whole human brain stem. So we can manage to get from centimeter to millimeter to micrometer and see whether the phosphorylation was on the Lewy neurites here or the Lewy bodies in the arrow head here. So it's kind of the first that we can do radio histological imaging because the sizes kind of, we bridge the scale from micro scale to macro scale with insights. And of course, if you are interested in whole mouse brain staining, we have you covered. We have a whole mouse brain here, like imaged in 2-photon tomography. So you can render it in terms of like a galaxy. And you can see the staining is very uniform. So you get absolute quantification of all these calcium binding proteins here. And we particularly choose these dense targets is because they are challenging. You usually don't get deep penetration for these densely expressed targets because they trap the antigen so fiercely. But now we can just get them all into the whole mouse brain. And here the staining was only performed for once. So the multiplexing was in a single step. So there is no difference from mapping a single marker or mapping 5 markers. It's just the same protocol. Okay. And of course, if we go to clinical tissues, we have prostate cancer biopsy here, like imaging the smooth muscle fibers and also some calcifications here. So we are trying to move towards the clinical realm. And if you want to scale across not the sample dimensions, but sample size, you can do it with multiple wells. So here is just staining all the 13 kidneys in parallel and then counting all the glomeruli. And the details are still preserved. So for example, for one of them, we have 4 channels here, and you can map all the tubules densely reconstructed in 3D. And this is in high contrast to iDisco. Even with iDisco for long staining times and lots of lectins added into it, you can see this severe rimming here, contrast to INSIGHT, which you can see the internal structures very clearly. So the effect is very dramatic. And I mean, for deep uniform staining is really, really uniform. So we can get to tracing single tubules across the whole kidney from all the glomeruli from the cortex to the medulla to the renal pelvis. So it's all going through, and there is nothing really to hide for this for all our images because all the signals are really there. There is no image adjustment needed for your imaging. Okay, and of course, if you work with embryos, we can have different stages of embryos that you can map for the neurovascular system in red and the nerve in blue. So you can it can even go through skull. If you decalcify the embryo, you can stain the whole brain, the central nervous system, the peripheral nervous system, and also the anterior nervous system. And we didn't punch a hole in the embryo, so the whole embryo is still intact. So you can stain your antibody through the skull, through the bone. And of course, we did a good rendering of the image because I mean it's pretty. So as demonstrated here, like the peripheral nervous system, you can see all the nerves are traceable from across the skull or just in front of the spinal cord. Some of you might recognize the familiar structures that you have never seen. And of course, something interesting is to look at the cranial nerve and the connection with the anterior nervous system like never before. So this one is around 1.5 centimeter and in size. And of course, you can fly through the whole image. And again, this image has not done any image processing. It's just rendering and without adjusting for the intensity much. So there is really nothing to hide. Yeah. And now if you want to scale across the color dimension, if you want to see more molecules, ever more molecules, because INSIGHT is a very minimally destructive technique. So you can even do this, you can even do RNA or DNA sequencing after doing the whole INSIGHT process. The sequence would not be biased or destroyed anyway. So even with conventional formalin fixed tissue, you can just dilipidate and then you stain with INSIGHT and clear it. After you image it, you can just elute the antibody with sodium sulfite. We detail it in our paper. Then you can go ahead to do another round of staining and imaging. And here we map 28 markers like here. And of course, the enlarged region would be more clear in terms of the, for example, the neuropeptide fibers in dots and vessels and GFAP, those kind of things. And of course, if you want to do high dimensional clustering, you can just segment the cells to localize all the 25 proteins and their 3D coordinates and also get the morphology as a bonus. So we can kind of go ahead to see what it can possibly achieve as because I'm also a clinician, I want to see what it can achieve in human tissues. So for example, here is a first time that we can map a 3.2 mm thick human cerebellum. So usually back in the days, we can only map like 200 micrometers. So it's like 16 times less thick. But now we can see the whole thing in its full glory and gory. And even the dense antigens like GFAP, neurofilament are like mapped throughout the whole thing. So again, this is a slice through here, and you can see the bubbling barriers formed by the astrocyte in cyan wrapping around the red vessels. And of course, you can go ahead to do a 3D analysis as usual. Okay, so again, another slice through. So there's again nothing to hide because all the signals are really there. And of course, because the signal is homogeneous, we can try to do some analysis that is only possible for homogeneous staining. So one of them is called structure tensor analysis. So it's a kind of an MRI analysis method that requires all the signal to be uniform throughout the thickness. Otherwise, you can't get the fiber tracks tracing their orientation, coherence, and something like that. So this is the first time that, again, we can try to do molecular scale diffusion tractography from each molecules like NFH and GFAP. And if we zoom in the details, we can do some structure analysis. Like here, we can visualize individual astrocytes at the bottom and how they wrap around the vessels and the Bergman glia on the molecular layer and all the neurofilaments and the relationships with them. And of course, we discover some new structures across the whole folium because we found these unusually thick neurofilament inclusions. We can't find any reference or literature describing them. If you found them, just let me know. We want to know what they are. Just that they are very, very prominent in 3D, but in 2D probably they would be disregarded as dust. So we map 1,700 of them here, and we are now trying to understand what they really represent. And another case that we have did is to try to analyze and deconstruct the whole kidney glomerulus. So we have an image stack of the whole glomerulus, and then we stain it with different markers, and then we segment it with AI. So here is the result. For example, a glomerulus here, and we segment all the different structures and reconstruct it in 3D. And we go ahead to analyze the structure function correlation. So we get the ground truth capillary areas and the filtration area fraction. Again, we computed the topological paths, and then we do hemodynamic modeling. Like we model the hydrodynamic pressure, the protein concentration changes across each point in the vessel, and also the ultrafiltration rate. So these are all biophysical modeling that is made possible by the real images. And then we went ahead to compare across the healthy control mouse glomerulus and the proteinuric mouse, and we found some topological defects in the lupus nephritis mouse model. So there is some mismatch in the lobule distribution that we are trying to understand mathematically what that means. Basically, there is a mismatch between the perfusion and the filtration. But if you are interested to explain more with us, just let us know. And of course, if we try to understand the phase space or the response space of the different glomerulus, we can find that their functions are very, very distinct in terms of their performance for the glomerular filtration rates and also the pressure difference. So somehow they are performing differently in terms of their functionality. So that would be only possible with this kind of detailed and also non-destructive 3D reconstruction. And of course, we are trying to move forward to the human one. Human ones are much more difficult because it's much larger, and there is a kind of chamber here that helps to distribute blood all over the larger arteries here. But we still managed to do the reconstruction because the staining was uniform and also the sample was quite intact. And here is the whole glomerulus reconstructed. So it's a scroll through, allowing you to see through all the different vessels and the filtration fractions in green. And of course, you can slice it in whatever way you want. So this one will be very helpful for clinical settings because sometimes you get ambiguous structures like this. And of course, again, with AI, you can do segment anything and get the blood vessels out. Along with the cells and then again, different channels. So those are quite basic for us already. And another thing that is complicated about humans is that we often make unexpected discovery because there is so little we actually know about them. So for example, it was assumed that all the glomerulus will have one input artery called the efferent artery, like AFF here, and also one output artery called the EFF, efferent artery. But it turns out when we map the whole thing in 3D and reconstruct all the paths of the connections, all the capillary connections, we found that there is actually one efferent and 3 efferents, so 1 input and 3 outputs. And we come to think of it, our textbook understanding on the glomerulus is really based on 2D sectioning, and then trying to mentally reconstruct it in our heads or by like a experience. And you can never get a case of perfect sectioning that you can get 2 or more efferent arteries on the same plane as the first efferent artery. And we verify that by doing another simulation to see that whether they are topologically distinct and they really perfuse different lobules. So in humans, there might be a separate mechanism that they can selectively perfuse different lobules to control the filtration rates in the kidney and control the rates of urine making. So this one is really kind of unexpected until we map the whole glomerulus in its entirety. And another area of interest is about liver. So we always wonder whether the liver has real sonation patterns, and that is very, very controversial over the last 50 years. So we really try to do a 3D H&E stain that we develop again, and also with 3D IHC membrane marker, so we can segment with ground truth each of the hexagonal hepatocytes. So we managed to analyze 50,000 cells. And the first thing that we discovered is that it is widely known that liver cells can have 1 nucleus, 2 nucleus, but we found that there can be 3 nucleus or even 4 nucleus. So here is an example. We just scan through the whole thing, and we will be able to find rare events. So here there is clearly 4 nucleus with crammed within the same liver cell. So it already broke the previous records. And if we try to analyze the sonation patterns, we can do this kind of a 3D distance transformation and see the higher ploidy, like the higher number of nuclei, is actually located in the central zone rather than at the periphery zones. So this actually kind of solves the debates that whether the higher ploidy will locate on the left side or the right side or the middle one. We will get to see the data later on. So what we did is that we do a simulated cut. So we take the whole 3D volume and then we just cut it in any kind of planes imaginable. And it's like a Monte Carlo simulation. And for that one, we try to quantify the membrane marker expressions, for example, DPP4, and then whether there is a signal per surface area expression pattern. So we see a clear bimodal distribution. So one population would be higher in DPP4 per surface area, and this second one would be lower. And when we do the same thing for 2D sectioning, then this time the metric will be like DBP4 expression in signal per perimeter. So it's one-dimensional less. So even with similar number of cells, we still don't get the bimodal distribution for a clear cutoff. So this kind of illustrates that even if you sample extensively for across the whole volume, if you are very, very hardworking, you can't see the pattern that is clear in 3D, but not in 2D. So it's like you are kind of constrained to being a three-dimensional being. You can't imagine the future and the past in terms of the fourth dimension. So there is a fundamental limit to traditional histology. And of course, as promised, here, for example, we finally resolved that the higher body is limited to the central zone of the liver, the zone 2. And if we try to do the same thing with 2D analysis, because of the readout of the vague readout, whether we are certain about whether a cell has 2 nuclei or more, so you can't get these kind of stratified layers of nuclear ploidy for the hepatocytes if we do a 2D simulated cut. So again, these are some of the features that we exemplify. You can't be sure with 2D, but when you just map the equal volume of 3D, everything will fall out clearly. So I think we have a lot of information today, but to summarize is that sometimes if you have a high dimensional view, it will allow you to, the first one is to quantify it much more easier if you know what you are looking for. And if you are trying to resolve a mystery, it allows you to give you a fresh perspective to investigate what you try to demystify. And then the third one, of course, is to try to discover what is previously unsuspected, like the kidney, new artery, or the 4 nuclear hepatocytes. Those are clearly unexpected. And I believe there will be much, much more to be discovered, and you can be the first one. And all these kind of images are produced by INSIGHT with the same single protocol without any modifications. It's just the same solution and with different antibodies. So we believe it is a very user-friendly method for every lab with basic equipment. Even high school students who know how to hold a pipette can do it. So thank you very much for your attention. Of course, all the credits goes to my my lab members. They are very helpful in doing all the hard work. And of course, our collaborators and colleagues, but most of all is the donors for the donation of the organs and also the tissues. And of course, our cat who always try to accompany us or try to destroy our work by deleting data. Yeah. And so thank you very much. I'm happy to take any questions if you want.
Thank you so much, Dr. Lai. It's very impressive technology, and we want to use it right now for different applications. So probably I will start on my side with some questions. So probably the first one at the practical level, where we can have your reagents.
Yeah, so we really receive a lot of requests from fellow colleagues. So we are setting up a commercial entity called Illumos. So you guys can go to the Illumos website or just Google INSIGHT. It's spelled wrongly for your Googling. Then you can just buy our kits or request a free sample, something like that one. Yeah.
Okay, thank you. And just in line with this question, at the technical level, so what kind of equipment is required to image in 3D with this kind of such depth of analysis?
Yeah, that is also a great question. So usually, if you have a confocal in your institute, you can already image a cleared 3D stained tissue. So in terms of like 1 millimeter thickness or lower. But if you really want to image larger samples, then you will need a light sheet microscope. We are happy to borrow you one or help you to image some of those samples. You just mail us the samples, then we will help you to image them. Because we are kind of passionate in doing this.
Okay, thank you. Yeah. So what kind of challenge, next challenge you expect or you want to take with your technology?
Yep. So of course, we are thinking about the future in terms of more colors, because our customers and also our collaborators always ask for more multiplexing or even spatial transcriptomics. So because INSIGHT is the only technique that can preserve RNAs in 3D, and so we are thinking about whether it can be compatible with spatial transcriptomics. It has some interesting result already, so we will be quite happy to share as soon as we have it. So that would be the future in terms of 3D spatial multiomics for us.
Okay. And so you discussed a lot about neurobiology. So do you have in mind other applications that will really benefit from this technology?
Yeah. So definitely we try to move forward to a clinical setting, because for diagnosis, usually pathologists are still limited to 2D and one single stain at a time. So they can't see much and they have to do a lot of guesswork. So we try to move that to clinical for cancer diagnosis to make them sure that the margin is clear, there is no cancer cells in the area, or the cancer cells are of high risk. So when they see more, they can be more clear about what they are dealing with. So we're trying to develop a diagnostic test here.
Okay. Probably I have another question, because we have a lot of interest in the field of oncology, in the characterization of the tumor microenvironment. And we know that organization of immune cells, fibroblast, endothelial cells are really important in the tumor progression and resistance to therapies. Do you believe that it can so your technology can allow to detect all this cell subset at the same time and in 3D?
Yes. Immunology is really the way, a very hot topic now. So yes, I think definitely for clinical samples, if we can get a lot of them, we can map all the immune cells in there in terms of the major markers, and segment all the cells and do a kind of a virtual flow cytometry in 3D space. And we have actually some data that tells us that the neighborhood, the spatial neighborhood of the immune cells actually would influence the outcome of the cancer patients, particularly those receiving anti-PD-L1 immunotherapy. So those kind of things, we are kind of planning a clinical trial on doing that. But it really see whether we have good collaborators to do it as an international trial.
Okay, great.
Yes.
Thank you so much.
Thank you so much.
So we do not have online questions. So I think we are now at the end of this session. Again, really thank you for this nice presentation. Lot of data, lot of interest. I'm sure, at least from our side, we are really excited to see all this data, to see our antibodies working in your hands. So thank you very much, and so we keep in touch, of course.
Okay.
Have a good day. Bye bye.